Hi, I'm Aaron Hosak, and I'm a senior in the Chemical and Biological Engineering Department here at CSU. And I'm proud to introduce the team in charge of the scaling up the COVID-19 vaccine production at this year's E-Days. The team members are Lindsay, myself, Sawyer, Mehdi, and Jack. So I'm going to go ahead and hand it over to Mehdi in order to talk a little bit about the introduction and background of our senior design project.
Introduction and background information. COVID-19 vaccine production scale-up is essential for vaccine delivery to both the developed and developing world. Despite the rollout of vaccines, cases continue to rise in both the US and globally. Our team looked at two prominent COVID-19 vaccine types as potential options, mRNA and adenoviral. The decision was made to focus on the scale-up of an adenoviral vector vaccine, similar to that of AstraZeneca and Johnson & Johnson. COVID-19 advector vaccines function through the recombinant insertion of an antigenic SARS-CoV-2 spike protein encoding plasmid into an adenovirus vector. The scale-up of adenoviral vector production involves the propagation of recombinant advectors in cell culture, followed by a primary isolation process and purification steps to eliminate biological and chemical contaminants. The safety, efficiency, and consistency of the scale-up process, as well as the efficacy and safety of the product, and economic feasibility of the production are all key design considerations. Here we see a simplified schematic of a typical advector scale-up process, taken from figure 10.16 of Prasers and Santos. The process begins with the introduction of a small amount of recombinant adenovirus containing the intended transgene, in our case, the SARS-CoV-2 spike protein plasmid. The adenovirus is then propagated using an appropriate cell culture. After this, a primary isolation step isolates the vectors from cellular components and other coarse contaminants present in the cell culture. An intermediate purification, involving precipitation, enzymatic treatments, or filtration steps, among others, then occurs, followed by a final chromatographic purification intended to capture, polish, and formulate the final product. The timing of the process depends greatly on the choice of cell line, as the cell density at the time of propagation is an extremely important parameter. The process in total takes hours to days to complete, and can result in upwards of 10,000 viral particles per cell for higher yields. Market aspects. Our team carried out a market analysis of the COVID-19 vaccine rollout worldwide. Key players in the international market include Pfizer and Moderna, who are producing mRNA vaccines, along with Johnson & Johnson and AstraZeneca, who are producing adenoviral vaccines. Of particular interest was the supply of vaccines to the developing market, in which both infrastructural and political challenges to vaccine distribution offer unique market opportunities and considerations. The stability of advector vaccines as DNA vaccines, which allows for higher carrying temperatures, together with the fact that prominent adenovirus vaccines only require one dose, offers a distinct market advantage in their supply to the developing world.
Three of the more significant decisions for the project were determining our target market, which vaccine types to use, and which cell lines to use to propagate the vector. The two target markets we considered were developed countries and developing countries. Developed countries can afford a higher priced vaccine, but the major vaccine players are well established and make competition difficult. While they cannot pay a high price for vaccine, the large unmet demand for vaccines in developing countries make them a more desirable market. With developing countries as our target market, the freezing temperatures needed for storing mRNA vaccines become prohibitively expensive, especially in remote areas. Adenoviruses are time-tested and more stable, and are the more reliable choice for developing countries. To render adenoviruses less harmful to patients, replication mechanisms in the adenovirus are impaired. So for successful propagation of the adenovirus, cell lines that can complement the deficiencies for the virus must be utilized. While Chinese hamster ovary cells are a popular mammalian cell line with extensive documentation, they lack the necessary replication mechanisms to complement the adenoviruses. Human embryonic kidney cells, HEK, and human retinoblast, PER, cells do have the necessary replication, and since the HEK cells have better documentation, the group used them for our cell line.
This process has four main steps to it. Those steps are seen right here, when the steps are inoculum preparation, cell culture and virus propagation, primary isolation and intermediate purification, and then the final step is final purification. This is the first step that we just talked about. As you can see, there's a little bit more in it than one single step. There's a couple air filtration systems, as well as mixing, gas compression, there's vessel procedures, all that kind of stuff. This is the second step of 4 steps that we were talking about. The first part of this would be blending and storage, which gets sorted into 2 dead end filtration systems, which then get put back into a blending and storage, which then goes to another blender, as well as split into an air filtration system. As we can see on this slide, there are 3 storage kind of mixers that we see, and we also see that there's a nucleus treatment tank and a centrifugation. These are just to help further the process and get it to what we need it to be in order to inject it into a human being. The final step in this is the purification. As we can see on here, there's a chromatography thing, a stoichiometric reaction, as well as ultrafiltration. These then go to a gel filtration, and then it gets filtrated again. And this is all just to make it able to go into our patients.
One important decision we had to make when considering our senior design project was whether to self-synthesize or purchase adenovirus. So the synthesis of our vaccine requires 2 starter adenovirus sequences. This consists of a helper and a gutless sequence. The helper sequence contains everything needed for it to both encapsulate itself and create the viral proteins needed for encapsulation. The gutless form only contains the Psi sequence, which allows it to encapsulate if viral proteins are present. Otherwise, it's completely empty. But what we did is we are able to fill that empty space with the DNA sequence for the spike protein from COVID-19. So our product adenovirus will have that spike protein and will induce an immune response within the patient. So these 2 adenovirus sequences can then be co-transfected into human embryonic kidney cells to produce recombinant or inactive adenovirus loaded with the spike protein DNA sequence. The process for how the vaccine-ready virus is synthesized is outlined in the diagram below. The issue we had is the beginning part of the process, we have a lot of unknowns. So we are not sure what the helper sequence actually looks like, nor the gutless sequence. We know that both of the sequences are transformed by E. coli and propagated, but then the process by which those are purified into the helper and gutless adenovirus are unknown. However, after that point, after we have the helper and gutless adenovirus, we know that the human embryonic kidney cells are co-transfected, and then those go through a final purification to make the recombinant adenovirus with spike protein sequence. So we know how those work, but due to the unknowns, we had to come to a decision whether to purchase or to try to figure out the process ourselves. Ultimately, we decided that the best decision would be to purchase the adenovirus starters. So we're going to purchase the gutless and helper adenovirus produced after the E. coli DNA transformation and propagation step. So and there are a few benefits to this. So we only need a small number of viral units to propagate the human embryonic kidney cells. And the adenoviruses, the starter viruses, are already commercialized, so purchasing them would be very simple. We do discuss this later, but we believe that investment into research of this process could be profitable, though. It would allow for development of a booster vaccine, since our vaccine uses adenovirus serotype 26. With our own technology, we could make a booster with adenovirus 35. Also, adenovirus delivery methods have potential in gene therapy, so we could potentially dip into other fields and create more profit that way.
As for future process goals, many aspects of the system would need to be investigated before vaccine distribution and optimization. The first order of business would be to fully specify the process. The skeleton process has been created. However, things like reactor kinetics, heat and mass transfer, and cell growth have not been added into our model, which would be a crucial part of understanding how the system reacts and would give us a better model of how it would react in the full scale production process. The next point would be to optimize the system through economic analysis. Aspects such as process time, energy prices, and starting material prices would weigh in on how we would produce the vaccine and how much of it we would produce, and delegate research toward other aspects to optimize the system. After full scale-up and full modeling of the process, FDA approval would begin. This portion of the distribution process would include things like clinical trials, verification, validation, and qualification activities. After FDA approval, distribution can begin, and after distribution, efficacy of the vaccine would be continually monitored, as well as the state of the production equipment. Adjustments to the process would be made as necessary. Process optimization and future research goals are desirable because they can maximize productivity or efficiency, as well as lessen overall costs to the company. A part of the future research delegation would be going towards creating an adenovirus starter. Because we are buying the adenovirus starter that we put into the system, it's a lot of upfront costs for the company. However, if research could be delegated towards creating our own, in the end, it would pay off. Another starting material that is bought for the process is the spike protein DNA. If it was possible to isolate the DNA within our manufacturing facility, it would reduce costs going towards the purchase of this DNA. Down the line, it also might be economically advisable to add on other processes to reduce starting costs of starting material. This would then, in turn, increase the profits for the company, allowing for delegation of funds to go towards other aspects of the drug, increasing its efficacy and safety.